Okay. Hi, everyone. We're excited to have Katie Young with us today. She's a postdoctoral researcher in Cynthia Reinert-King's lab at Vanderbilt. And I guess you guys are moving if you
We
haven't
are.
already. But we're excited to have Katie present to us today about the mechanical and genetic data at single cell level, which is also what she did her PhD on at Georgia Tech. So please take it away, Katie.
Thank you so much for that introduction. Yes, and thank you to everyone who's logged on or is going to watch this later. I'm really excited to share with you guys the work that I am doing here at Vanderbilt and will do at Rice, as well as some of the work that I had the opportunity to work on during my PhD with Todd Solchek at Georgia Tech. So I'll be sharing with you all about my work on relating mechanical and genetic data at the single cell level across the genome to investigate metastasis. I really don't need to spend a long time belaboring the point that cancer is not good and metastasis is much worse. My work has focused on both ovarian and breast cancer, which together will lead to tens of thousands of deaths this year alone, a majority of which are caused by the metastatic spread of tumor cells throughout the body. To successfully metastasize, a small subset of primary tumor cells undergo several phenotypic changes to survive the many steps of the metastatic cascade. The subset of cells from the primary tumor that can survive all of these changes is unique and thus could be uniquely targeted to stop metastasis, which leads to a really big question. What exactly are the mechanisms that drive this metastatic subset of cells? While there are many ways we could view this problem, I am particularly interested in thinking about the physical properties that change during the metastatic cascade. So during each of the steps of metastasis, cells undergo these big physical changes when they rearrange their cytoskeletons and generate force to move through the tumor microenvironment and survive various forces that are imposed on them throughout circulation and while crawling through confined spaces. For this reason, as this group knows well, we are really interested in studying the mechanical properties of cells, such as how stiff or soft they are, and how these properties then affect different biological processes. The Solchek lab, where I did my PhD, has demonstrated, along with many other groups, that for many cancer types, not only are cancer cells softer than normal cells, but metastatic cells are softer than their non-metastatic cancer counterparts, with these softer cell types having increased migratory and invasive abilities. When comparing the stiffer ovarian cancer cell line, HEY, here circled in green, with the more metastatic and softer daughter cell line, HEY A8 in purple, we had observed a population-level gene expression difference, which revealed increased expression of cytoskeletal remodeling proteins in the more metastatic population. This initial population data led to the question, can we relate gene expression to cell mechanics? In order to fully capture this relationship, we really need to drill down to the single cell level. There are tens of trillions of cells in a human body, but it is the mutations in specific programs in individual cells that lead to tumor formation. Bulk analysis approaches will take millions of cells and provide average values of cellular heterogeneity, or of those cellular characteristics, missing out on all of that cellular heterogeneity, and also losing information about rare cells that could actually be driving tumor growth or metastasis. Single cell techniques, on the other hand, allow us to understand these differences between cell populations to identify which subpopulation is truly driving malignant behavior. Several other studies helped demonstrate that there's this importance of and the ability to link cell transcriptomics with cell function and phenotype. However, there had not been a study linking single cell mechanics and gene expression prior to, I'd say, my work and study that resulted in my PhD thesis in 2021. The main objective of my research is to understand the molecular and mechanical mechanisms that drive metastasis. And I hypothesize that there's this link between cell softening and a more mesenchymal migratory phenotype that is controlled by the expression of a network of genes of interest, which are advantageous during the metastatic cascade. Today, I'll be specifically talking about 3 projects that I've been working on to explore this central hypothesis. First, I created and validated a combined single-cell mechanics and gene expression methodology in order to look at the single-cell relationships between cell mechanics and a subset of genes of interest. Next, I expanded my search using a genome scale CRISPR gene editing scheme, while in conjunction with this high throughput mechanical screen, to generate more hypotheses on this link between genes and mechanics. And finally, I've been currently working on studying the stability and heritability of cancer cell mechanotypes in order to separate stiff and soft breast cancer cell populations, which I will then use to study the effect of cell mechanics on each of the stages of in vivo metastasis. The first project I'll talk to you today about is where I developed a new method to combine single-cell mechanical and gene expression data. This was driven by the hypothesis that there are correlations between gene expression and cell mechanics that can help us investigate different cells' metastatic potential. In this new single-cell genomechanics method, I first grew cells on gridded cover slips, which allowed me to keep track of the spatial location of each cell of interest. I analyzed images of the cells and calculated several shape descriptors, such as cell elongation and cell solidity, which helped capture the presence of cell protrusions, which are usually characteristic of a more mesenchymal cell state. Next, I used atomic force microscopy, or AFM, to measure the mechanical properties of individual ovarian cancer cells. I developed, along with some other lab mates, some custom code to extract the cell stiffness and cell viscous properties from large numbers of AFM force curves at once. After collecting the mechanical data for each cell, I used a micropipette setup to aspirate and isolate single cells using negative pressure to slurp up each cell of interest, and then positive pressure to then place it in an individual tube of lysis buffer for downstream analysis. Using this method, I collected over 300 single cells from my 3 cell types of interest. Now that I collected the mechanical data and actually have the cells collected, the last step was to look at the genes of interest. I used a multiplex targeted quantitative PCR approach, which allowed for the analysis of 96 genes in 96 individual single cell samples at a time. I curated my list of genes of interest from an extensive literature search, including reviews of genes related to metastasis, cytoskeletal remodeling, genes differentially expressed during EMT, and differing expression between HEY and HEY8 cell types. This new method allowed me to generate a large linked single cell data set to investigate the correlative relationships between mechanics and gene expression. Before we really dive into that single cell information, I noticed several interesting relationships at the population level for our 3 cell types of interest. The more migratory and mesenchymal cell line HEY8, its parent mesenchymal cell line HEY, and the epithelial and less migratory OVCAR3 cell line. On the right, these rain cloud plots allow us to observe how each single cell measurement, which is depicted by each dot on the plot, contributes to the population distribution, summarized both by that box plot and the half violin plot above. With increasing migratory ability from the OVCAR3 cells to the HEY8 and HEY, we observed both cell softening and changing viscous properties for the fast and slow viscous time constants. Similar, although not identical, to some of that data I showed earlier from former lab member Wenwei Zhu. With our morphology measurements, we observed that as cell migratory ability increased, we saw increased cell elongation and a reduction in cell solidity, which is associated with more protrusions, a characteristic of more motile cells. Looking at the gene expression information, I observed several interesting population trends. With increasing migratory ability, we saw an increase of expression of pro-metastatic genes, such as EMT-driving transcription factors, extracellular matrix remodeling proteins, markers of cancer stemness, and mesenchymal markers. while we saw the opposite trend with decreasing expression of anti-metastatic genes, such as epithelial markers, metastasis suppressors, cell differentiation markers, and ECM remodeling inhibitors. When comparing the average gene expression of individual cells as compared to bulk gene expression levels, we confirmed the two measures were significantly correlated, implying that the isolation method had little effect on the measured gene expression. Importantly, we also observed the population measurement represented as a point here in the violin plot did not actually represent the true expression of many actual single cells and did not accurately capture the heterogeneity of that single cell gene expression, further supporting our use of a single cell measurement to investigate the intersection of cell mechanotype, genotype, and phenotype. To actually look at the large linked data set, we observed thousands of significant pairwise correlations between our genes of interest, our 3 mechanical parameters, and 7 morphology descriptors. Here I've plotted all the significant correlations, with red representing positive correlations and blue representing negative correlations. There really is a wealth of information that we could explore here about how different genes are expressed and in related manners and the effect of this expression in different cell types. But to start, we wanted to investigate the relationships between specific genes of interest and cell stiffness. We identified significant correlations between single cell Young's modulus, or cell stiffness, and the gene expression for 47 different factors. Diving deeper into these relationships, we can group these factors by their functions. 10 of the 14 negative correlations are associated with this pro-metastatic processes, meaning that higher expression of these genes is associated with a softer mechanotype, while 17 of the 25 positive correlations with cell stiffness are associated with these anti-metastatic processes. I was really interested in several of the specific genes from the analysis, such as the negatively correlated transglutaminase 2, a cross-linking protein involved with extracellular matrix remodeling, as well as migratory and stem-like properties, the intermediate filament vimentin, an important mesenchymal marker and marker of EMT, ZEB1, a transcription factor implicated in the EMT process. Of note from the positive correlations were the metastasis suppressor and metalloproteinase inhibitor TIMP3, the nuclear structure protein lamin A, which protects against DNA damage and provides a major barrier to cell invasion, as well as E-cadherin, an adhesion protein associated with an epithelial phenotype. These genes of interest further support this hypothesis of an association of a softer mechanotype with a more migratory metastatic phenotype, while also providing further jumping off points to explore the molecular mechanisms and gene networks that govern this cell softening phenomena. In addition to understanding these genomechanical relationships, we were interested in seeing how each of these data types that we collected contributed to our ability to distinguish between groups of cells of different metastatic potential. We conducted a principal components analysis to reduce the dimensionality of this large data set, clustering similar cells together. Looking at gene expression, mechanics, and morphology data alone was insufficient to separate cells by cell type, which I was using as a proxy for their metastatic potential. I was able to separate the epithelial and mesenchymal cell types pretty easily with just looking at genes alone, but where the method really struggled was in separating the HEA and HEA8 cells, which are both mesenchymal in nature, but differ in their metastatic ability. However, combining multiple data types using partial least squares analysis followed by PCA revealed an ability to better separate these cell types, with the best clustering observed when combining all 3 data types, which even allowed for that separation between the HEA and HEA8 cell populations. The Davies-Bowden index is a measure of success of a clustering algorithm where a smaller value of dB is more optimal. By minimizing the differences between or differences within a cluster while maximizing the difference between the groups, we were able to see this reduced dB index after combining multiple data types, and which was indicative of this optimal clustering, further supporting this need for these linked data sets whose collection was enabled by this new technique. So in summary, for this first project, there are a couple key takeaways. First, we observed that a softer cell mechanotype, mesenchymal morphology, and pro-metastatic gene expression were all observed with cells with increased migratory potential. Second, we identified several genes related to this metastatic process that were correlated with stiffness. And finally, we showed that combining these multiple data types can improve our ability to separate cells of varying metastatic potential by cell type. To move on and shift gears a little to this second project I'll talk with you about today, I used a genome scale CRISPR knockout or GECKO strategy in concert with a high throughput mechanical screen to further identify genes related to the mechanical change throughout the genome. I hypothesized that there were certain gene knockouts that would be significantly enriched in the stiff and soft outlets of our microfluidic cell stiffness sorting device, which would further inform how gene expression changes play a role in the cell softening and stiffening process. This GECKO mechanics method starts with a lentiviral library of 76,000 guide RNAs, targeting the knockout of all 19,000 protein coding genes in the genome, with 4 to 6 guide RNAs per gene. A Cas9 expressing cell line was transduced with the lentiviral library, and antibiotic selection was used to isolate the cells with successful single gene knockouts. By using a low multiplicity of infection, statistically, you're able to get about one gene knockout per cell. After creating this library of cells with single gene knockouts, I was able to use our lab's microfluidic device to sort cells based on their stiffness. After the sort, the genomic DNA is then harvested and sequenced to determine the post-screen guide RNA distribution. Guides against genes that affect the cell softening and stiffening processes will be enriched in those different outlets and then provide further hypotheses about relationships between genes and cell mechanics. To introduce you all to the microfluidic cell sorting platform, the technology consists of a microchannel with multiple diagonal ridges that hang down from the ceiling of the device and compress cells as they flow through the channel. Highly deformable cells, denoted by the circles in this cell trajectory here in red, are able to squeeze underneath the ridges and follow the hydrodynamic forces of the surrounding flow buffer. So these softer cells move straight through the device. Alternatively, stiffer cells resist compression and are moved by those diagonal ridges perpendicular to the fluid flow. With the device, we can sort cells based on several biophysical properties, and our group has demonstrated the ability to fractionate cells based on cell viability, chemoresistance, stem cell differentiation, and malignancy, even from ovarian cancer patient samples. So before I started taking on this really large genome scale project, I needed to answer the question of whether these single gene knockouts would be enough to drastically affect the cell stiffness. My first indication that this might be possible was looking to the original paper validating the device efficacy, where former student Billy Wang showed the ability to sort cells that were artificially softened with cytochalasin D, which resulted in a 50% reduction in cell stiffness. I compared this stiffness difference to that of a stiffness change of a single gene knockout. I had contributed some work to it for another publication where we knocked out PTP in 21 and then measured the stiffness difference. So as you can see, the knockout of this gene in hematopoietic stem cells resulted in a similar reduction in the cell's Young's modulus as the sortable cytochalasin D treated cells, helping confirm that this large undertaking could be feasible. I next took some AFM measurements of cells transduced with the GECKO lentiviral library, and I observed significant change in the population, both in the mean and in increased variation in cell stiffness, observing at least 5% of these knockout cells exhibiting stiffness higher than that of the maximum measured control cell stiffness. I next wanted to confirm that the GECKO cells would behave differently in the microfluidic device when compared to cells without any gene knockouts. We were able to track our cells in the microfluidic device with a custom Heimdall Python package the generated cell trajectories help us understand how these different populations of cells interact with the device, as well as predict our sorting ability. And yes, in case you were wondering, the Python code for this project is named after the Marvel character Heimdall, who sees all. Tracking the gecko and control cells in our device, we did see increased deflection and spread of the gecko population cells, especially at the first ridge. This data helped support moving forward with this project, demonstrating our ability to fractionate cells by their mechanics after single gene knockouts. Here, however, was where I first experienced a pretty major hurdle to the project. To maintain a required coverage of approximately 500 cells per guide RNA, I needed to sort around 40 million cells. So previously, the throughput of our sorting device was limited to approximately 240,000 cells per hour for optimal sorting. But to process the tens of millions of cells required for the sort, I would need to run the experiment that original device for over 8 consecutive days. I spent a lot of late nights in lab, but this schedule was untenable even for me and definitely for my cells. Therefore, I needed a major redesign to improve the throughput of our device. To do this, I had a couple knobs that I could turn. I could increase the concentration of the cells processed, but this was limited by cell-cell and cell-device interactions that start to happen when the density of the cells in the device are too high. Additionally, you could try and increase the velocity at which you were processing the cells, but this also was limited by the way that our sorting device operates. At higher flow rates, the hydrodynamic forces outweigh the cell elastic force interacting with the ridges, and all the cells would end up moving just straight through the device, regardless of mechanics, if that speed is sped up too high. A third option for increasing throughput is to parallelize the channels. By keeping the same optimal flow parameters, you can linearly increase throughput with every additional channel. So therefore, I redesigned our device for parallelization by first linearizing and then duplicating the sorting channels and implementing a manifold layer for the distribution and recombination of cells from similar outlets. So after many design iterations and fabrication in the clean room, I was able to assemble a multiplex device that could result in the even distribution and recombination of cells in 8 sorting channels with the same flow parameters, all while maintaining a simple control with only 3 inlets and 5 outlets, which was the same as the original device. For a closer look at this final multiplex design, we used 2 layers with a bottom layer with 8 parallel linearized devices depicted in green. We used an adjusted channel width to allow for equal volumetric output to each of the outlets. The top layer of the device has a manifold for even distribution and recombination of cells from the outlets after sorting. And we also used that adjusted channel width to allow for this even flow to and from this layer, which allowed for easier sample handling to avoid the use of a total of 24 inlets and 40 outlets. One of the final tests for efficacy of this new device was ensuring similar flow parameters in each of the channels after cell distribution. So using that cell tracking software again, we observed a slight variation in individual channel velocity. However, we used a heat treatment model system, which drastically stiffens cells, and we're able to observe that these differing cell trajectories between the soft viable and stiff non-viable cells across the range of these flow velocities depicted in this plot were seen in the 8 parallel channels. So after all of this optimization, we wanted to confirm that we could maintain sorting efficiency across each of our design iterations. Previously, our original tree design, we were able to enrich both live and dead cells in the soft and stiff outlets, respectively. We saw very similar sorting efficiency in our linear design. And although we saw a reduction of our enrichment of soft cells in our multiplex design, we were still able to highly enrich our stiff cell populations, creating these highly pure samples of stiffened cells. Most importantly, while our sorting ability remained approximately the same, the throughput of our device increased over 20-fold. To try to put this in perspective, the processing time for our project that requires 50 million cells was reduced from over 8 days of continuous sorting to just 9 hours. This enabling step allowed us to move forward with this Gecko mechanical screen. For the Gecko transduction process to work, we needed a cell line that stably expressed Cas9. We developed an ovarian cancer Cas9 GFP cell line. And when we used a guide against GFP, we saw a significant reduction of fluorescence of cells, as well as a 25 to 60% total insertion deletion efficiency at that cut site. We transduced these Cas9 expressing cells with the lentiviral library containing the 76,000 guide RNAs. I repeated the sort 4 times, sorting approximately 40 million cells every time, for a total of over 150 million cells processed with these single gene knockouts. As expected from the AFM data I showed earlier, there were around 3% of the cells that were deflected to those stiffer outlets in outlet 4 and 5. I harvested the genomic DNA and then sequenced the guide RNA region to create another really large data set for exploration. We were able to identify hundreds of genes that were significantly enriched and depleted across the 5 outlets. And from this larger list of enriched genes, I've developed a list of 26 genes of interest that have 100-fold enrichment, whose expression results in changes in cancer patient survival according to Kaplan-Meier curve analysis. Included on this list is CCDC88A, or GERDin, which is an actin-binding protein, which is important in Wnt signaling and cell migration. I'm continuing to process and interpret all of this sequencing data, and I'm excited to continue to uncover more of these interesting hypotheses about how genes are correlated with cell softening and cell stiffening. In summary, for the second project, a couple key takeaways. First, I was able to redesign this microfluidic sorting device to drastically improve the throughput and reduce processing time by 20-fold. I was able to mechanically screen 150 million knockout cells and generated an enormous sequencing data set, which I am continuing to explore for these potential genes controlling cell mechanical changes. The last project I'd like to introduce to y'all is some of the current work I've brought to Vanderbilt University, comparing these in vitro cell mechanical properties to in vivo metastatic ability. I hypothesized that by understanding how cell stiffness plays a role in each of the stages of the metastatic cascade in vivo, we'll be able to help find these therapeutic targets that could specifically change cell stiffness and then also metastatic ability. This is some work in progress, and I'm happy to share some of the advances that I've made to the cell sorting platform in order to create these soft and stiff mechanotypes of a breast cancer cell line, creating these different cell subpopulations. The inspiration for this project, and one of the reasons I decided to join the Reinhardt King Lab, was a paradigm shifting study that came from a previous student, Lauren Happich, who used repeated rounds of migration-based cell sorting through transwell assays to create this stable subpopulation of cells that were highly and weakly migratory in vitro. Interestingly, when these subpopulations of cells were injected orthotopically into mammary mouse fat pads, it was the weakly migratory cells and not the highly migratory cells which showed increased metastasis to the lungs and liver. Lauren then used engineered models of each of the steps of the metastatic cascade to identify that the main difference between the subpopulations in vivo was the ability of these weakly migratory subpopulations to form circulating tumor clusters in circulation, which led to more robust metastasis. This difference between in vitro migratory behavior and in vivo metastasis really made me wonder if we could observe a causal relationship between cell mechanics and in vivo metastasis. Many groups have noted this correlative relationship between cell stiffness and metastatic potential, but I'm really interested in studying how stiff and soft cells behave differently during each of the steps of the metastatic cascade. To do this, I will first serially sort these breast cancer cell lines based on their cell stiffness to separate the populations into soft and stiff subpopulations of cells. Next, both mechanotypes of cells will be used to form mouse models of metastatic disease, and I will investigate the role that cell mechanics play at each of the steps of the metastatic cascade and observe how cell mechanics change at each of these stages. And finally, a multi-omics approach will be used to investigate the differences of subpopulations at the transcriptomic, proteomic, and epigenomic levels in order to determine the underlying molecular mechanisms that link cell mechanics and metastasis. Sorting the cells in the microfluidic device, as I mentioned earlier, relies on this balance of hydrodynamic forces and normal forces experienced by the cells under each ridge. Therefore, the flow rate at which the cells are moved through the device affects the distribution of the cells sorting to each outlet, with the soft cells expected to be sorted to outlets 1 and 2, and stiffer cells deflected to outlets 3, 4, and 5. When optimizing and testing efficiency, I chose to test the mechanics of cells sorted at this 40 microliters per minute, where I first started to see deflection of cells in the device. I used some AFM again to measure the stiffness of cells going to each outlet. However, interestingly, I actually did not observe a relationship between cell stiffness and outlet when sorting at this particular flow rate. This led to the use of multi-stage sorting for the first time with this device. So I noticed that at this 40 microliters per minute, I actually had really great size-based sorting. And so after doing an initial size-based sort at this 40 microliters per minute, I took all of the size binned population of cells from that outlet 2, and then sorted those cells again at a lower flow rate. This stage sorting removed the effect of size on cell sorting, but I was actually still able to observe cells deflected to those stiff outlets at lower flow rates, implying I was no longer sorting on size, but rather on stiffness. My next step was to confirm this stiffness-based sorting. Because the cells are in suspension during the sorting process, I decided to use micropipette aspiration to study cell mechanics in suspension. Micropipette aspiration works by applying a ramping negative pressure to a cell and observing the length of the cell pulled into the pipette as a function of the negative pressure. Stiffer cells resist deformation and show a steeper relationship between aspiration length and applied pressure, while softer cells easily deform and aspirate into that pipette. Using the technique, I was able to demonstrate an enrichment of stiff cells in the 5th outlet after both size and stiffness staged sorting. So after confirming the enrichment of stiff cells after this stage sorting, I used a serial sorting method to develop subpopulations of cells with stable, stiff, and soft mechanotypes. To do this, after sorting a heterogeneous parental MDA MB231 breast cancer cell line, I took the enriched outlet 5 stiff population and allowed the cells to grow and divide before resorting the cells again, taking the stiff outlet 5 cells from that sort. This process was repeated again and again, taking the outlet 5 population every time for expansion. The same process was repeated as well for the enriched soft population I was collecting in outlet 2. This serial sorting process has been continued until I saw the proportions of cells sorting to each outlet stabilize. So after multiple rounds of serial sorting, I have indeed seen an increasing proportion of the serially sorted stiff outlet 5 population being sorted back to that outlet 5, as well as a similar trend for the serially sorted soft outlet 2 cells. This indicates that the mechanical property of cells can be inherited and is stable enough to see this population level shift in reaction to stiffness-based sorting, even after expansion and passaging. Interestingly, I have also began to observe some morphological differences between the serially sorted populations with the outlet 2 population appearing more mesenchymal and the outlet 5 population appearing more rounded. Even with the size-based exclusion staged sorting, I've continued to see a shift in cell size between these serially sorted populations, leading to some additional questions about perhaps an intrinsic link between cell stiffness and cell size. Here I've compared the strain energy experienced by each cell under the ridge, which is this product of cell volume, cell stiffness, and the strain experienced by each cell under compression. This metric helps demonstrate that there are these mechanotypic differences between my developed cell subpopulations. I'm really excited as I've sort of developed these different mechanotypes of cells to move forward. So in summary of what we've learned so far, this multi-stage sorting has allowed for this enrichment of high strain energy cells that are stiffer and larger, which has enabled the development of subpopulations with these different mechanotypes through serial sorting. I'm looking forward to further exploring the stability of these mechanical properties and discovering by which pathways this property is passed from cell to cell. My next step will be to use these developed cell lines to explore the effect of cell stiffness on the primary tumor escape, survival and circulation, extravasation, and colonization at metastatic sites in an orthotopic mouse model. So in review of what we've discussed today, I first investigated the intersection between cell mechanotype, genotype, and phenotype in 3 projects. First, looking at these single cell genomechanics method that I developed to look at the single cell correlations between cell mechanics and a subset of genes of interest. Next, I drastically improved the processing time for our cell sorting device so I could expand my search by using a Gecko mechanical screen to generate another large linked data set. And now I'm building off that work from my PhD by investigating the role of cell mechanotype that it plays in the in vivo metastatic cascade and in the molecular regulators of that process. I would love to give a really big thank you to everyone that was in the Solcheck lab and all of the collaborators and funding sources that I had access to at Georgia Tech, as well as a big thank you to all my current lab mates and current funding sources. With that, I would be happy to take any questions about any of the topics I discussed today. Thank you.
Great. Thank you so much, Katie, for a wonderful talk. If you have any questions, please feel free to unmute yourself and ask, or you can put it in the chat as well.
I have a question. How stable are the stiff cell types that you prepare from the microfluidic system? You may have said this and I missed it, but if you collect a whole bunch in the stiff channel and then you plate them and then rerun the assay, do they separate among all the channels again, or do they all go to the stiff one?
Yes. So great question. And I'd say kind of an area that we're continuing to explore. They do not all go back to the stiffest outlet after, but as I've been serially sorting these cells, the proportion of cells that return to the 5th outlet, it has increased. I think some of it, the ability to see all the cells go back to the same outlet is hampered by some of the efficiency of the device itself. So I personally think that with the efficiency, I will never see 100% of the cells return to the 5th outlet. And some of that is related to, like, as the cells are flowing through the device, there is some stochasticity and randomness of, like, if a cell approaches one of the ridges and there might be another cell that is nearby, then that might affect the cell trajectories and things like that.
Yeah. But you don't think it's because you don't think that the cells kind of revert back to some other softer phenotype after a cycle?
I think it might be a combination of both. So a project that I didn't discuss here that is sort of in its beginning stages right now is really kind of asking that question of, like, how stable is this property of taking AFM measurements of the same single cells over, I guess the pilot studies I've done so far have been, like, kind of over a day's worth of time. But I'm interested in taking that out multiple days to see, okay, if we take AFM measurements of the same cells at day 1, day 2, day 3, and then, like, look at the progeny of each of those cells as well.
Yeah.
Like how exactly, and then kind of getting to the question too of like throughout a cell cycle, how might that stiffness be changing, going up and down, and can we,
Right.
I think because of the serial sorting and that I've been seeing these shifts in populations, I believe that there's something that is allowing the entire population to shift in this way. But I think on the individual cell level, there's some flexibility in that mechanotype. And I'm really interested in that question, especially before we're going into the mouse model, where the cells will be experiencing all of these different mechanical environments. And my guess is we'll be changing their mechanics during that process as well.
Yeah, really nice.
Thank you.
Hey, Katie, thank you. That was a really awesome talk and impressive work. I was just wondering, because you mentioned how softer cells are better, are more metastatic, essentially. So I'm wondering, because we classically think of tumors as being stiffer.
Right.
So is there a relationship between the softness of the tumor and metastasis, or is this more of a single cell phenomenon?
Yeah. So when I first joined the lab during my PhD, and Todd said, "Oh, we see this relationship between cell stiffness and metastatic ability," I was like, "Oh, that's so surprising," because as a kind of naive first year grad student, I was like, "Oh, like when you think of cancer, you think of like hard lumps sort of thing." And so I think there's this really interesting relationship where at the kind of ECM level of tumors, you would see this like tissue level stiffening, while the cellular component, we're seeing this sort of softer mechanotype associated with more metastatic cells. So I'm interested in becoming a faculty member one day, and when I'm thinking about my future lab, I think that's a question I'm really interested in as well, of like how these like softer cells might be associated with like a stiffer environment, and how those two components of a tumor feed on top of one another. And thinking about sort of force generation of like how are these softer cells being able to maybe pull on the stiffer environment to move around and make it through each of those stages of metastasis.
Cool, thank you.
All right, are there any additional questions for Katie? If not, thank you all. Oh, there's one more in the chat from Kayla. Can you talk a bit more about the sorting for the Gecko project? It looks like you were getting far larger proportions of stiff cells and low proportions of soft cells into the channels. Can you explain why you were seeing that and whether that affected the data set?
Yeah, so I think I may have gone through that sort of distribution plot kind of fast. So actually what we were seeing is a large proportion of the cells go to that sort of outlet 2, which I classify as a softer channel. So this idea that a lot of these gene knockouts might be resulting in cell softening, and then and some of that is sort of the nature of the device, of the way that the diagonal ridges are set up. We're really good at enriching for stiffer populations. So we are able to get these like more highly pure stiff cell populations. And so when looking at the genomic sequencing and looking at the guide RNA distribution, the significant enrichments that we saw were really related to gene knockouts, which resulted in cells going to that stiffer outlet. And so I was actually pretty excited about the ability to pull out like which of these gene knockouts might be associated with cell stiffening, just from the sense that if we're sticking with this paradigm, thinking about cell softening being associated with more metastatic properties, being able to identify the genes that when knocked out result in stiffening, these might be some of the genes that are related to this cell softening process that we might want to therapeutically target. But it is a great question of like, just in the screen itself, are we kind of imposing some like kind of bias to the data that would be of like which genes we're pursuing. So now that the big screen is over, I am following all of the hypotheses up with some individual gene knockout validation to figure out, okay, which of these genes of interest might be worth pursuing more.